Hello there. This is learnsciencex.blogspot.com. Welcome again to our lesson series on transport, diffusion, osmosis, and plasmolysis. Today we are moving on to osmosis. Now, what is osmosis? Osmosis is the movement of water molecules from a dilute solution to a more concentrated solution across a semi-permeable membrane. Osmosis only deals with movement of water molecules from a dilute solution to a more concentrated solution across a semi-permeable membrane. The higher the difference in concentration between the two solutions, the faster the rate of osmosis. A rise in temperature also speeds up the rate of osmosis. Is that okay? Now let's look at something. So we said that osmosis is actually the movement of water molecules from a dilute solution to a more concentrated solution. We can have a beaker and then we fill it a bit of water over here. This one to the plus symbolize solute and the dot symbolize water molecules or solvent. This one in the middle here is the semi-permeable membrane. Now, this is solution A, this is solution B. Now remember, a solution is made up of solute and then we have solvent. Solute is any substance which gets dissolved. Is that okay? It could be sugar, it could be salt. We also have solvent. Solvent is usually liquid. Okay, so we have water as the universal solvent. Is that okay? Now, for solution A, we have only few solute there. So the plus symbolize the solute. For solution B, we have a lot of solute but few solvent. The dot is the solvent. Few solvent. So always for osmosis, it is the movement of water molecules from a more dilute solution. In other words, from a solution which is having low solute concentration. So osmosis could be defined as the movement of water molecules from a solution having low solute concentration to a solution having high solute concentration, which is the B, solution B over here. I hope we are getting it. Always in osmosis, water molecules are moving from solutions having low solute concentration to a solution having high solute concentration. In other words, movement of water molecule from a dilute. So this is a dilute because there is a lot of water molecules here. Dilute solution to a more concentrated. This is more concentrated because there is a lot of solute. The plus a lot of solute here through a semi-permeable membrane. That is osmosis. A semi-permeable membrane is a membrane that allows certain substances to pass through and blocks the passage of some other substances depending on the nature and size of the substance. Is that okay? We have impermeable membrane, which doesn't allow anything. Solute, solvent, it doesn't allow. Then we have semi-permeable, which allows solvent molecules to pass through, but it doesn't allow solute molecules to pass through. Now we have an experiment to demonstrate osmosis in a semi-permeable membrane. Peel off the skin of a yam tuber, cut it into two, and then you make a cavity at the ends or at the center of each half and flatten the bottom. Label one A and fill it with known volume of a concentrated salt solution. Label the other one B and fill it with water. Fill the two vessels with water to about 1/3 full and record the volumes. Sit the yam cups into each of the vessels. Leave the whole experimental setup for 24 hours. So this is the experimental setup. You have your beaker here. You have a beaker here. Peel your yam and then just chop the centers. Chop this one too. And then you fill it with water. And then you fill it with water. So this one is concentrated salt solution, while this one is just ordinary water. This one is water. This one is water. What are we going to see? Observation. After 24 hours, it will be observed that the level of concentrated salt solution in yam cup A has increased in volume, whilst the water in the vessel has decreased compared to B. The volume of water in both yam cups and the vessel will remain the same. So for beaker A, the concentrated salt solution will increase. For beaker B, it will remain the same. Conclusion: water molecules in vessel A or setup A have moved from a low concentration to a high concentration salt solution through the yam, which acts as a semi-permeable membrane. Don't forget that the yam is acting as a semi-permeable membrane. In setup B, the water molecules in both the vessel and the yam cavity have the same low concentration. Therefore, there was no movement of molecules from one region to the other. This is a very, very simple experiment. So the first one that I explained is the same thing. This one was having bond solute concentration. So water molecules will move from here and enter into this one. That's why we have had an increase in the salt solution. Now let us demonstrate osmosis in non-living tissue. We cover the ends of two tester funnels labeled A and B with cellophane paper. And then we fill funnel A with sugar solution and funnel B with water. We climb them both upside down into a beaker of water. We leave the setup for 24 hours. Now observation. After 24 hours, it will be observed that the level of liquid in test tube A would rise, but that of test tube B would remain the same. So conclusion: Water molecules have moved from a region of lower concentration to a higher concentration through the cellophane paper, which acts as a cellophane as a semi-permeable membrane. This is the setup. This one also is a very simple experiment. Alright. Now, example of osmosis. The first one is absorption of water from the soil by roots of plants. The next one is movement of water across the cells of plants. The next one, we have movement of water from the root hair into the cortex. Then we have absorption of water in the colon or large intestines of mammals. Then we have absorption of water in the nephron of the kidney of mammals. The last one, we have entering of water into the cells of amoeba. Now let's look at some differences between diffusion and osmosis. So the first one, diffusion occurs in both gases and liquids. Osmosis occurs in liquids only. Diffusion does not require partial membrane. Osmosis requires partial membrane. Diffusion involves movement of any substance, both solvent and solute. Osmosis involves only solvent particles, example, water molecules. Alright, so we've come to the end of this lecture. Now there's a project here, so you can try it in the house, or if you are a group of students, you can try it. Use the principle of osmosis in the following activities. Number 1, salting of fish for preservation, example, tilapia or Kobe. Number 2, preservation of liquid food products, example, fruit juice concentrate. Don't forget to keep on practicing until we meet again. Adios for now. This has been learnsciencex.blogspot.com. Don't forget to subscribe to our channel, YouTube channel, and whatever. All right.